Okay, our final speaker for the session is Dan Gibson. So in case model and non-model organisms didn't cover enough territory, so we now have synthetic organisms. So it's an honor to introduce Dan Gibson. Dan is Vice President of DNA Technology at Synthetic Genomics. He is best known in our community as the inventor of the eponymous Gibson assembly for DNA. Dan developed Gibson assembly at the J. Craig Venter Institute, where he co-led the team responsible for constructing the first bacterial genome in 2008, and later the first synthetic cell in 2010. He'll be telling us about design and synthesis of cells from the bottom up.
Thank you very much. Now for something a little bit different, I'm going to go through my presentation without saying CRISPR. So the long-term vision of my groups, both at the Venter Institute and at Synthetic Genomics, is to develop the capacity to design and build synthetic organisms from the bottom up. So ultimately, we want to have genome design software such as this, where you can select all of the characteristics that you want in your cell, and then the genome design software will give you that complete genome sequence, which you can then synthesize and activate to make a self-replicating cell. And we want that genome design software to be predictable, so that any changes that you make at the chromosome level can then be computationally modeled at the systems level. So we believe that technology such as this is really going to be essential to fully understanding biological systems, so that we can then begin building next generation production platforms for medicines, biochemicals, biofuels, nutrition, agriculture, anything that you can think of. But the problem right now, the challenge that we have, is we don't yet understand how to design and build a cell from the bottom up. So unlike electromechanical devices that are designed by engineers, which have a rational and modular architecture, genomes are constructed by evolution. And so they are seemingly irrational, untidy, and highly improvised. Our goal is to change this. Okay, we want to rationally design a genome from the bottom up. We want to get rid of all of the stuff that we don't need, and all the stuff that we do need, we want to modularize that. That's our goal. And in order to get to that point, then we need to first establish some very basic genome design principles that we can then plug into this genome design software. In order to test those genome design principles, we need to learn how to chemically synthesize a chromosome, and then we need to learn how to activate that synthetic chromosome to make a self-replicating cell. So this is exactly what my teams, first at the Venter Institute and later on at Synthetic Genomics, have been working on for the past 10 to 15 years. So here's an overview of our approach to building a synthetic cell, starting from DNA sequence information. There were really three major breakthroughs. The first breakthrough was learning how to assemble very large pieces of DNA in vitro using an exonuclease, a polymerase, and a ligase. I called it the one-step isothermal assembly method. I'm not responsible for calling it Gibson assembly, but that's what it's currently known as. The second breakthrough was finding that we could use yeast to very efficiently take up and assemble very large pieces of DNA where those pieces of DNA could be recombined into a complete bacterial chromosome. And to that extreme, we've actually been able to assemble whole genomes from 24 overlapping DNA fragments that were each 24,000 base pairs in size. And the third breakthrough was finding that we could move those whole chromosomes out of yeast into recipient cells and reprogram those cells based on the DNA sequence that's coming out of those yeast cells. And in some cases, even converting one species to another species. So all of these technologies that we've been developing for whole genome synthesis and activation all came together back in 2010 when we announced the creation of the first synthetic cell. This is a cell that naturally has a very small genome at only 1.1 million base pairs based on Mycoplasma mycoides. So ever since Craig Venter and his team first sequenced a complete bacterial genome back in 1995, thousands more whole bacterial genomes have been sequenced, stored in databases. And our work was really the control experiment of what we ultimately want to do. So we were able to reverse this process, proof of concept that we can reverse this process, pull a whole genome sequence out of the computer, chemically synthesize a structure in the laboratory, then activate it through genome transplantation to make a synthetic cell that's based on that genome sequence in the computer. This work, although it gave us an extraordinary set of tools for whole genome synthesis and activation, it really didn't teach us much about genome design. And that's because we really relied on what nature had already given us, an existing DNA sequence. Sure, we made a few edits to it, but in the end, it really was no surprise. We pulled out a sequence of Mycoplasma mycoides out of the computer, and we built Mycoplasma mycoides. But what this work did give us was this whole design, build, test cycle at the whole genome level, where we could now, with our genome synthesis and activation technologies, we could start testing numerous design objectives and begin addressing a wide range of fundamental biological questions. One of the questions that we've always had, first at the Venter Institute and later on at Synthetic Genomics, our teams wanted to understand how we could build a minimal cell that has only the core machinery necessary for independent life. So we imagined building a cell that has only the core functions. These would be the essential genes that make the cell alive. And then we would have adaptive functions which would enable growth of the cell in a variety of environments. The reason why we'd want to do this is because it's a really nice opportunity to investigate the first principles of cellular life, where we could begin understanding and defining what the role of every single gene in this minimal cell is. And then we could begin computationally modeling the cell. And also, it could be used as a launching pad for learning how to design and build more complex organisms, and possibly being used as a chassis for industrial applications. So let me just very quickly, I'll take you through our design, build, test cycle for how we go from, how we went from the Mycoplasma mycoides synthetic cell, which we called SYN 1.0. It has a 1.1 million base pair genome, and how we ended up with a minimal cell. So our design was based primarily on TN5 mutagenesis, so that we could begin defining essential, non-essential, and quasi-essential genes. And because we wanted a cell that grew at a reasonable growth rate that we could actually use once this project was completed, we decided to keep all of the genes that are both essential and quasi-essential. So ever since we've constructed the first synthetic cell back in 2010, we've made some major advances in our genome construction technology, which really allowed us to accelerate this design build test cycle. So this included integrating some of our genome construction processes on SGI DNA's BioXP, improving the speed and fidelity at which we can build these constructs by adding error correction procedures, and also doing as much of the amplification of those constructs, including very large constructs, in vitro, and also developing alternative host organisms for cloning these DNA constructs. And so I'm not going to walk you through this now, but I'll be around to answer any questions you have about our workflow. And so test. This is a process that was developed by John Glass and Carol Lartigue through genome transplantation, where we pull the whole chromosomes out of yeast. We put them into recipient cells from Mycoplasma capricolum, and then we see if the donor genome can survive and give colonies after approximately 2 to 3 days following the genome transplantation event. So following this design build test cycle, we had to go through 3 iterations of this, and we ultimately got to our minimal cell, which we called SYN 3.0. So shown in blue is the SYN 1.0 genome. It has a genome size of 1.1 million base pairs and has 901 genes. And the SYN 3.0 minimal genome has a genome size of only 531 kb and 473 genes. So we've been able to remove over 400 genes while maintaining a growth rate of less than 3 hours. So to put this into perspective, Mycoplasma genitalium has the smallest genome of that's known in nature, has a genome size of 580 kb. So we've been able to build a genome and show that it's functional, that when it's 50 kb smaller than that, that's found in nature, and it grows about 5 times faster. So as you would expect, the minimal genome retains all the essential genes known for known core cellular functions. We put these into 4 categories: expression of genome information, preservation of genome information, cell membrane structure and function, and cytosolic metabolism. And there are 79 of these genes that we couldn't assign to any functional category. And if you look at this blast map of proteins in the SYN 3.0 genome compared to homologs found in other organisms, what we did here is we grouped the genes that we were very certain about on the left to all the way to the right, where we literally have no clue what the function of that gene is. But what's interesting is, although we don't understand the purpose of that gene's function, we know that it's absolutely essential for life, and it's also conserved along a variety of other organisms. So this is part of what we see as a really nice opportunity to start defining in a very simple system what are these genes of unknown function. We started out with about 149. I believe we're about less than 100 now. Another one of our long-standing goals has been to organize the genome such that all of the biological processes are grouped together. We refer to this as either modularization or defragmentation, to use a computer analogy. So far, we've been able to show that we can modularize approximately 12% of the minimal Syn 3.0 genome. We published our minimal cell paper last year in Science. This is an effort that began over 20 years ago by Ham Smith, Clyde Hutchison, and Craig Venter. I was fortunate enough to work with them on this project over the past 12 years. So to summarize this part of my talk, the creation of the Syn 3.0 minimal cell resulted in robust genome construction workflows and a chassis for scientific discovery. It's because of these genome synthesis and activation tools which are now enabling the discovery of genome design principles for minimization, modularization, and novel gene function. We always imagined, once we got to this point, that we would use the minimal cell as a launching pad for making more complex and useful organisms that can be used in industry. I believe as we gain a deeper understanding for how to design life from the bottom up, we'll get to this point. But for now, a faster and more cost-effective approach is to start applying our synthetic cell and minimal cell technologies to a more evolved system that already exists in nature. And about 3 or 4 years ago, we found that organism. It's a gram-negative bacterium called Vibrio natrigens. We call it Vmax. And this is why it's an ideal starting point for us. It's not a pathogen. It can be genetically engineered. It has a fast growth rate. In fact, this is the fastest growing organism that's known in the world. It has a doubling time of less than 9 minutes. It grows to high density and low cost medium. It grows in defined medium, so you can then therefore begin better defining the complete genetic system. It has the potential for recombinant protein secretion. It has natural transformation machinery, which allows you to accelerate some of the genome engineering efforts. And it's already compatible with many of the E. coli protocols, vectors, and reagents that are already entrenched in just about every molecular biology laboratory. So over the past 3 to 4 years, we've been developing a very comprehensive molecular biology tool set for Vmax for molecular cloning and protein expression applications, and this work is being led by Matt Weinstock, shown here in the red shirt. So here's a growth curve comparing Vmax in blue to several E. coli strains, including one of the fastest growing E. coli strains shown in gold. So you can see Vmax grows much faster into a higher density than any of the E. coli strains. And on the right is showing that Vmax colonies are clearly visible after 6 hours. You can even see them after 4 hours, but none of the E. coli colonies are yet visible. A major reason initially for bringing Vmax in-house was to accelerate some of our laboratory workflows, so specifically the cloning process. So most of you are familiar with this, where you assemble your DNA fragments and then you transform them into a host organism. You then have to wait for colonies to appear on plates. And then you have to pick those colonies, grow them into liquid medium, and allow the cells to grow to a high enough density where you can then purify your plasmid DNA. So with E. coli, this is traditionally about a 3-day process, but with Vmax, it becomes now a 2-day process easily, and if you're very aggressive or have very aggressive grad students, you can carry this out in a 1-day process. So In order to get to this point, we've had to develop a very comprehensive molecular biology tool set for Vmax. So we've established efficient methods for transformation and isolation of DNA. We've developed plasmids that have various origins of replication and selectable markers and even counter-selectable markers, both for cloning and protein expression applications. And we've shown that Vmax is compatible with our DNA assembly methods and readily available growth medium. We've also established protein expression capabilities in Vmax. This is, we've been able to induce proteins by either IPTG, arabinose, or temperature, but then applying those systems to the more commonly used T7 expression system. We have that system working really well to express not only green fluorescence protein, as you see here, but also a variety of other very diverse targets. One of the things that many of you may be familiar with is if you try to express proteins in E. coli, some of those proteins are toxic to E. coli, or the proteins are not toxic, but they make the cells very sick, or that protein is expressed, but it goes into the insoluble form. What we're seeing with our early data with Vmax is it seems to capture a wider range of proteins that can be expressed and are found in the soluble fraction, which can really help facilitate the downstream processing of those proteins. What we're seeing compared to E. coli is you get about a 3 to 4 times increase in biomass, and which is equating to about a 3 to 4 x increase in protein yield, and this is both in comparing experiments either in shake flasks or in fermenters, and this is summarizing some of the key features comparing Vmax to E. coli. The most significant being the doubling time. You get your experiment completed faster. The cells will even grow pretty quickly at room temperature, an increase in biomass, an increase in protein, overall protein yield, and the potential for proteins being more soluble. You can purchase now these Vmax protein expression strains through SGI DNA. So if you talk to me, I can tell you more about that. One of the other things that we're very excited about with Vmax is if you look at the complete genome sequence, it has multiple secretion systems, and unlike E. coli, it very clearly has these type 1 and type 2 secretion systems, which allow you to express and transport your protein and secrete it directly into the growth medium, which can really help facilitate the downstream processing of that protein. So we've moved now beyond just a bioinformatics exercise. We've shown that if you grow the Vmax cells in the presence of sucrase, or sucrose, there's an enzyme called 11-sucrase, which is secreted, which makes sense because that's the enzyme responsible for breaking the sucrose down into glucose and fructose. So we've been able to now separate out this induction system where we can put the 11-sucrase gene under the control of the T7 system. So this is a step toward getting to where we ultimately want to go, is having Vmax be a plug-and-play system for recombinant protein secretion. So this is what's currently in the works with Vmax. For reasons that I've already mentioned, and because the design build test cycle is accelerated using Vmax. We're using Vmax to develop our bacterial genome design software. We imagine building a Vmax genome that's completely minimized, modularized, and recoded. So some of the benefits from an industrial applications point of view, with minimization, you remove pathways that either compete or interfere with the desired process. With modularization, this allows you to upgrade a complete biological process all at once instead of doing edits here and there all around the genome. So it can really accelerate the genome engineering efforts. And to recode the genome would give you a genetic firewall against phage infection and from horizontal gene transfer. So we're thinking about, now we're in the process of building Vmax as an optimized chassis for biotechnology. And just really thinking about these cells that are very specifically tailored to the production of, whatever you think of, whether it's insulin or what, just very, very efficient systems where you have, they're dedicating all their energy to a particular process, and there's little or no byproducts. I don't have a functioning timer up here, so I don't know how much, am I out of time? Okay, I'll wrap up then in 3 minutes. So just the final part of my talk really is just a couple of slides to show you that we're not only thinking about genome design software of prokaryotes, but also eukaryotes. So our go-to organism for this work is Cliveromyces marxianus, or what we call Kmax. It goes with the Vmax Kmax theme. Cliveromyces marxianus is the fastest growing eukaryote. It's a thermotolerant yeast that has a very small 10.9 million base pair genome spread across 8 chromosomes. And a major difference between a prokaryote and a eukaryote is with a eukaryotic system, you have the post-translational modification that you would not have in Vmax, for example. Our approach goes like this, thinking about the design build test theme, where with the design is based on comparative genomics of dispensable genes and other yeasts, using transcriptomics and proteomics, and also random mutagenesis, where we can identify genes that are dispensable, but you also don't sacrifice the growth rate. Our build process is to build these chromosomes individually, and then and we've already actually built one of these chromosomes, chromosome 7, shown here, 930 kb. And then our test approach is similar to the genome transplantation work that I discussed, only it's not a whole genome at a time, it's one chromosome at a time. So you take your synthetic chromosome, swap it in, do a replacement, and ask if the cells are viable. So this is new work that's only recently got started, but we're thinking about minimizing and defragmenting a complete Kmax genome. So I just want to conclude by letting you know that many of the tools and reagents that I've discussed throughout my talk are commercially available through SGI DNA. If you want to check out the website for more information. Many of you may be interested in the Vmax cloning strain, which will be available later this year. The protein expression strain for Vmax is already out. with that, I want to thank the great members of my teams at JCVI, Synthetic Genomics, and SGI DNA. I've already acknowledged Ham and Craig and Clyde, who are our genomics pioneers. So with that, thank you very much. I'm happy to take questions.
So the Syn3 genome contains these 149 genes, about 30% of all, that are mysterious in a sense. So I'm thinking about a possible caveat here of the following kind. Could it be the case that some of these genes are not actually essential for growth, but you cannot remove them because another gene with which this thing interacts becomes toxic? Have you considered this? They're antitoxins, so to speak. Have you considered that possibility, or the possibility to remove bigger blocks of the genome that might help?
Yeah, I think that's a very interesting thought. You know, we'd have to go back to the data because, as you just pointed out, I mean, we do have lots of different combinations of genes that, in the process of doing this, that we have. So we would need to look at that and see if there are any cases where these genes that we're calling indispensable are indeed dispensable under certain conditions. That's an interesting thought. I'd have to give that some more thought. I'm happy to talk with you further about that. Thank you.
So it was a fantastic talk. And first, I wanted to thank you on behalf of everyone in my lab and myself for developing your reaction, because we use it every day and it makes everyone's life easier. So cheers. So in the context of the defragmentation, I'm wondering, I mean, it seems like there has to be some reason that those things have been arranged, or some efficiencies that have been developed across evolution. And have you noticed when you do that, there are some genes that don't like it, and for whatever reason, crowding or regulation in the 3D geometry of the genome, that become problematic?
Yeah. So let me just say, with our Syn 3.0 minimal genome, we took 1/8 section and we said, let's just focus on this one section, let's modularize it and see what happens. And we were shocked that it worked. So we're 1 for 1. I think as we move forward and try to understand rules for doing that, I believe that we're going to see some major issues with doing that are going to probably be some new genome design principles. I'm guessing that that's going to be more straightforward in a prokaryotic system like Vmax. I think we're going to end up seeing more problems with Kmax. I mean, you might be familiar with some of the work that Chris Voigt did, for example, where he was modularizing a genetic circuit for nitrogen fixation, and that resulted in less fixation of nitrogen. So I believe we'll run into some unexpected problems, but that's the purpose of doing this work, is to define what those rules are.
Thanks.
I want to say it's always been a fan of the work. Actually, got a really quick question. Just how difficult is it to do the genome transplantation in the Vmax?
We haven't pursued genome transplantation in Vmax at this point. I think, but it's interesting that you mentioned that, because it does. And one of the things I didn't mention is that Vmax has a 2-chromosome structure. It has a 3 million and a 2 million base pair chromosome. So what would be interesting is if we could build one of those chromosomes perhaps in a reduced or significantly engineered form, and then do a complete swap of one of those chromosomes at a time. Genome transplantation, there's lots of considerations, including how compatible is the recipient cell. Are there restriction modification systems? You have to have the hardware of the recipient cell established to recognize and boot up that donor genome. So we're not at the point of doing that with Vmax yet, but this is probably a challenge that we're going to face once we get to that point. Thanks for your question. Okay.
All right, so let's thank Dan and all of our speakers for the whole day.